Our last speaker is Vida Mingo from Columbia College. Vida, could you share your screen, please? Hello, Vida.
Yep, so I'm here sharing.
Perfect.
Oh, okay. So sorry. It was sharing just fine. Okay, there we go. Can you see it?
No, I can't.
Okay, all right. Might have to. Yeah, just practice the sharing. It's working just fine. So sorry. This would happen to me. But it's all right. Had it up here. Oops, so sorry. I'm sorry. Wow. I might need a bit of assistance because I shared it earlier. All right, I'm gonna do this.
Vida, did you have a question? Do you need some help or?
Yeah, I had it shared earlier. I just had it, I had it open and now I'm going to share and it is not currently sharing, which I'm like, oh gosh, this is, yeah, it's not actually good. It's actually.
Did you want to Alt tab or Command tab over to the Zoom session, and then down at the bottom there's a green share screen button.
Yeah, I have the share screen. I'm sharing it, and it's not what I'm finding. Wow, that's
Fabulous.
There we go.
okay. So sorry. So now that I've kept you all in suspense, but essentially here I am, Vida Mingo from Columbia College. Sharing, as you can see here, I want to first highlight my co-authors, as you can see here. So to kind of talk about the Genomics Education Partnership, it's quite fitting. We had mentioned from Sally Elgin, as well as the introduction earlier about cures. And as you can see here, we have the GEP. We are gracious and thankful to be funded by the NSF and the NIH. Any of the materials that I'm going to present and talk about today can be found here at gep.org. So overall, one of the things we want to do in GEP is to really just to introduce this idea of genomics and bioinformatics. As many of the other presenters have talked about, is one of the things that's critical about bioinformatics is, particularly now that we're in a COVID environment or without, is the fact that it is low cost. It provides a level of accessibility for students to be able to actually engage and to engage in authentic research projects. So as you see here, one of the things I would like to point out is the number of actually genetics faculty that we have. Also, and other speakers have talked about, the level of inclusion that can actually occur with actually using this particular type of bioinformatics cure. So as you can see here, a number of our minority serving institutions, historically Black colleges and universities, first generation, non-traditional, commuter, as well as residential. So it really gives you an idea of the span in the reach that we can reach. As you can see here, this is just representatives of individuals from our particular partnership, as you see here with Washington University. So ultimately, as you're going through this annotation process, one of the things we are setting students up to do is to be able to identify, to locate, and understand the overall process of science. And so we can use this to explore any number of different tools about, particularly here, about evolution. And as you can see here, this is just one example of a genome browser. As you can see here, where a student has this model, and then this is actually gene predictors. And so students are able to really get involved in the actual process of science. This idea of going through the data, exploring the data, coming up with a model that they can actually defend. And as you see here at the bottom, making an original contribution. But essentially at the very top is really what we're looking for students to do, to actually create this gene model. What you see here is the University of California, Santa Cruz, the Genome Browser. This is just one representation of the Genome Browser. There are other tools here that I just didn't show, Flybase, the other processes that you would see. And as you go in, whether it be Gene Record Finder and how students are able to take these, one of the things that's critical is students have to look at varying models. Sometimes they may conflict, they may contradict, and students have to come up with a gene model, but particularly to be able to defend their conclusions, to really be able to communicate the particular process of science. And this becomes really important. As you see here, I hear and I forget. But one of the critical things we like to focus on in GEP is this idea that, what you do is what you understand. And so you learn science by engaging in science. And so these are some of the skills, as always been mentioned already, about entering the scientific community, about building a knowledge and talking about this analytic thinking and how to use comparable lines of evidence. We have designed this based on a curriculum, again, that you can find at thegep.org, where you have curriculum for beginning students. As you see here, whether it be through modules, I've used this with my own students to kind of take them through the modules so they can really take that particular dive in. You can start here. Again, we have videos that are associated. And this is a process where we would like to engage students, but also, retain them as they move through the process. So as you can see to the right, you have, advanced curriculum for those students. Once they become common with annotation, we can move them on into the actual process. And so this is one of the first projects that we have here with the F element. So as you see, at the top, we're using melanogaster as our reference sequence. And this particular process, we have D-ananasi. And one of the things that became really surprising in looking at, as you see here in this actual paper, is this particular expansion. This 14.6 expansion was something that was really remarkable. And so as you can see, the publication that's at the bottom that actually talked about it. And as earlier was mentioned about the number of co-authors. So as you see there, you have 239 co-authors that actually participated as students. So this is actually getting students involved in original genomics research. And as you can see, the total amount that was actually done by the work of these students. Another project that we have here is the actually parasitoid wasp project. This is with Nate Mortimer at Illinois State and others that are working. And so what we're really focusing on here is trying to understand more so about the host and the parasite, interaction, but particularly looking at this role of conservation. And see, as you can see at the bottom right, is this really looking at these annotation of these three different models. And next is the next project that we have is the Pathways project, as you can see with Dr. Laura Reed at the University of Alabama. And so really looking at the very top here, you're seeing that looking at this protein evolution, and it seems to be highly conserved actually at the top, and particularly looking at insulin signaling pathway. So over there you see particularly in this role of Chico and the role that it's playing. And as you're looking at this signaling pathway, you really be able to answer this particular question as well as others. So as you can see, this project will certainly expand in the future. If you are interested in any of these projects, you're more than welcome to contact myself or anybody on the author list. Shifting gears to kind of really hit home this idea of who we are as a national organization. As you can see here by this particular this graph here that's actually showing who we have from 2006 is when it was started, the University of Washington, St. Louis. into how we spread all across the country. One of the things I really want to hit home is this distributed leadership model that we use at GEP. So we have this model, and it's actually broken up into actual regional nodes. And again, we've gotten funding to show this. As you can see on the left here in bold are the number of actual regional nodes. So if this is something that you're interested in, please, by all means, contact any one of us. I have highlighted the Carolinas node that represents North and South Carolina, and that's the node that I particularly work in. And so I've talked a lot about the process, but what are students saying? Because this is really the overall goal. This is student-centered and student-focused. What you're seeing is just a few examples of what students quotes. And so earlier somebody was talking about this logical thinking, analytical reasoning, and you can see one of the greatest votes of confidence is the words of our students. Particularly, I would like to highlight the student that talked about that aha moment. As educators, that's one of the things we are always looking for, that light bulb, that aha moment that helped to propel each of us to become the science and the educators that we are today, particularly defending a hypothesis, which is something that we all know is necessary in science communication and the budding of a young scientist. And so as you see at the bottom here, from a faculty member. So this is this interaction. We know one of the greatest impacts on a student is faculty interaction. So having faculty that are engaged in this process with students in this level of discovery, it has tremendous gains on the students. But not for necessary to just take my word about it, as this is science education. So what are some of the things we've learned? As already been mentioned, bioinformatics is low laboratory costs. We have web-based tools. We have this, publicly available data that students can use. There's not the same lab safety issues. You can access it 24 hours a day. So if you're like me, with my campus, we have a lot of commuter students or students who are living in rural parts of our state. If they have the bandwidth, they're able to do this. It lends itself to actual peer instruction, which I've actually used. Mistakes are inexpensive in terms of time and resources. If a student made a mistake, you're able to go back and look at it and actually kind of talk about it and break it down. We're able to, we've done research to look at in terms of gains, whether in terms of belonging or even in terms of research gains. And so we've compared that against, as you see here, against the Sure. But one of the things I did want to, certainly focus on is this concept of formative frustration. This is one of the things that we certainly wanted to highlight. And so in looking at this idea of formative frustration, this is what you're seeing here. The citation is here on the right if you are interested in looking more information. But really this idea of a student is grappling with a project, they're wrestling, and they may become frustrated. But it's in this process that students are able to find their voice and to be able to have that aha moment. So this facilitating growth through frustration. One of the things we're really trying to put forward is there is a level of growth that occurs. I've seen it with my own GEP students. She struggled, but as she got more comfortable and was able to manage the tools, she was able to say, I was really able to see this. She really was able to see how it came together, and she really took ownership of the learning on her own. And so with that, again, I would like to thank my co-authors. Again, I'd like to thank the NSF and NIH, and I'm certainly open for questions. Thank you so much.
Thank you so much, Vida, for your presentation. So I encourage folks to put their questions in the Q&A box. I'll start off with a question. So what kind of time commitment or what kind of context would you be implementing this kind of a cure in?
Yes, I would say one thing, it varies. It depends on what the particular faculty member is looking to, whether it be they have it as a standalone cure course, or what I've done is done it as an independent study, so I have it to where I have it set up. I actually met with the student, for about, 2 hours or so a week. They have particular assignments, and then we kind of work through the modules, and then they have a particular project. And so again, it depends on the faculty member. You can set this up for an entire semester. you can set this up for X number of weeks. It just depends. One thing is interesting is it's customizable depending on the faculty member.
So we have a question about what kinds of prerequisite courses would you recommend for this cure? Biology, math, chemistry, that sort of thing.
Yeah, so I would certainly say, you have to have like a biology 110, foundational biology, certainly molecular biology, just so that students have some idea of the central dogma, a genetics course, and, certainly if there's, if a student has access to computer science, that would actually be useful. But if not, I've done it with students that haven't had any, computer science background. Like we don't have evolution at my actual particular institution. So I was able to kind of fill, a lot of that in. But yeah, I would say certainly genetics and molecular biology, which is generally standard of most biology courses.
So most folks would do this. The students would be sort of sophomore, junior,
yes,
or senior. All right. Okay, so if we don't have any other questions, I'd like to thank all of the speakers for their presentations today, and thank you everybody for attending this workshop. And I hope to see many of you at some of the other education and undergraduate type programming for the rest of the conference. So if you're on the East Coast like me, go have some dinner.
Thank you all.
Thank you.
Thank you.
Thanks, y'all.
Thanks, everybody.
Going to bed now.
Bye.